Good morning. The topic for today is the genetic code. The genetic code is a set of rules by which the information which is encoded in the genetic material, that is DNA, is translated into the language of amino acid, that is, it is translated into a protein molecule by living cells. The information in DNA is in the form of triplet codons. Now, what is a triplet codon? The triplet codon is made up of 3 bases. So, as you can see in the figure, codon 1 is made of 3 bases G, C, U. Each of these codons will specify a particular amino acid. So, the codon, the genetic information which is present in the DNA is first transcribed into mRNA and then it is transcribed into proteins. And as I told you, every triplet codon, like for example, codon 1 will specify for a particular amino acid, codon 2 may specify for another amino acid. So every triplet codon in the DNA will specify a particular amino acid in the protein. The DNA has a 4-letter language. The 4-letter language comprises of the bases A, T, G and C, that is adenine, thymine, guanine and cytosine. Translation involves the conversion of this 4-letter language of bases into the 20-letter language of amino acids. Now, as you can see here, this is the DNA template, and each codon is made up of 3 bases, and it is first transcribed into the mRNA molecule. So these are the complementary bases. For AAT, for example, the complementary basis is UUA, and for this particular codon which is present on the mRNA molecule, the amino acid which it specifies is leucine. So a triplet codon is one with 3 bases, and this codon is sufficient because there will be 64 combinations by permutation and combination that you can have. It can code for 64 different amino acids. Since there are 4 bases, 4 to the power of 3 is equal to 64 different triplets of bases by permutation and combination. In comparison, a doublet code, so if it is only a doublet code like AA or the corresponding bases in mRNA, UU coding for a particular amino acid, then the possible combinations are only 16, and this is not enough to cover the 20 amino acids that we see in protein molecule. So the genetic code is a series of codons, and it's read one triplet at a time. Now, certain amino acids may be specified by more than one codon. Out of the 64 possible codons, 3 of these codons are the nonsense codons or the stop codons. They are UAA, UGA and UAG. The remaining 61 codons specify amino acids. So some of the amino acids can be specified by more than one codon. So for example, in the case of leucine, as you can see in the table here, leucine may be specified by CUU, CUC, CUA and CUG. As I told you, the Terminator codons are 3 in number, which does not code for any particular amino acid, and therefore they are called as nonsense codons or Terminator codons. The Initiator Codon is usually AUG. It acts as the Initiator Codon and also the codon for methionine. In few cases, GUG may be the Initiator Codon. Now, what are the characteristics of the genetic code? Firstly, it is degenerate. There are 61 codons for 20 amino acids. So, certain amino acids, as I showed you earlier, may have multiple codons. For example, for serine, there are 6 codons. For methionine, there is only 1 codon. For tryptophan, there is only 1 codon. Now, the third nucleotide in the codon is less important, and this accounts for the degeneracy. It is unambiguous. That means if you are given a specific codon, only a single amino acid is indicated. It is non-overlapping. The reading of the genetic code during protein synthesis does not involve any overlap of codons. The codons are consecutive. Next, it is not punctuated. Once the reading is commenced at a specific codon, there is no comma or full stop. It is read continuously till the nonsense codon or termination codon is reached. It is almost universal between species and between tissues within its species. So why is it almost universal? Because there are exceptions. The mitochondria has its own translational mechanisms, and certain codons are read differently in mitochondrial protein synthesis. For example, AUA codes for methionine instead of isoleucine, CUA codes for threonine instead of leucine, UGA codes for tryptophan rather than the stop codon, AGA, AGG serves as stop codons in the mitochondria. Then there are alternate genetic codes for ciliated protozoa. So the genetic code is almost universal, but it is a standard genetic code. Now the degeneracy of the genetic code, as I told you, resides in the last nucleotide of the codon triplet. So there is base priming between the last nucleotide and the corresponding nucleotide of the anticodon is not stable. It is flexible. Okay. For example, arginine, it has two codons, AGA and AGG. The third nucleotide in case of the first codon is A, and the third nucleotide in the second codon is G. The first two bases are the same, A and G, in both the codons of arginine. But this is recognized by the anticodon UCU. So the anticodon UCU recognizes both the codons of arginine, that is AGA and AGG. So the explanation of this wobble was proposed by Francis Crick, and therefore it was known as the wobble hypothesis of Francis Crick in 1966. This is to explain how a single tRNA molecule can bind to 2 to 3 different codons. Now the essential features of his hypothesis are: first, the first two bases in the codon form standard Watson-Crick base pairs with the last two bases in the anticodon. But there is a certain amount of play or wobble or flexibility in the base pairing between the first base of the anticodon and the third base of the codon. This permits the non-standard base pairing. When the first base of the anticodon is C or A, the base pairing is very specific, and only one codon is recognized, as you can see in the table here. But when the first base is U or G, the binding is less specific, and two different codons may be read of the codon and the first base of the anticodon. So in the first figure, you find that there is a normal base pairing between G and C. It's a normal pairing, normal Watson and Crick pairing. Whereas in the second figure, you find that there is a wobble because the third base in the codon is U, and that is base pairing with G. So CUU and CUC of leucine is being recognized by the anticodon GAG, and this explains the wobble hypothesis of Watson and Crick. Thank you.